Yes, I mean, perhaps I should say that if you have any interest after this, the best place to look is on the Nobel Prize website, where they now have, after a couple of years, my autobiography, along with the lecture, a written version of the lecture that I gave, which is
I've read it 3 times
to get through. Right? Okay. Well, you are going to interrogate me then. So that's definitely that's probably the best place to look to give a flavor of some of the things I've done that might interest you the most. But if I go back to those things which sort of, that you've suggested I mention, I was actually brought up in West Africa. So I was a premature baby, and my parents were worried that I would die if in England they weren't allowed to have any extra coal. It's a bit like the vaccine saga now, where even if you're very vulnerable, you don't necessarily get it because you don't fit their parameters. And so my father, who was having difficulty getting a job anyway, went out to West Africa, and apparently after that I thrived. So that was within my first few months of life. And so that was in the Gold Coast, which then later became Ghana in the late 50s, early 60s. And so much of my life the early life was in Ghana, and I was, of course, surrounded by wild. So I learned lots of hymns. We never learned anything else. And when I had to come back to England and was exposed to English country songs, I hadn't the faintest idea what they were talking about. But I did know the hymns. In those days in primary school, which is where I spent my time there, it was a university primary school, and it was very interesting in the sense that they forged a link with the University of Ghana, or the Gold Coast then became University of Ghana, and this little school was based in a sort of outshot of Accra called Achimota, a village, which at that stage a big forest around it, and great big snakes lived in the forest and such like. But in the school, it was in a little oasis there, basically in the woods, with a field, and it was a sort of mission school. And we went there for the morning, so we got up early, between about 7 and about 1, I think, was our day. Then we came back home. My mother was made sure we did our homework, because if we did our homework, then in the afternoon at 4 o'clock, we could go to the university pool where we could go for a swim. So that was our life. So we had these 2 hours of having to do homework and doing it as fast and efficiently as we could. And if we didn't do it well, then we didn't go. If either of us didn't do it well. So my brother and I, who was about 1.5 years younger, had to make sure that we kept an eye on each other to make sure we were working, because we could both suffer. And, but of course there was time in afternoons when we couldn't go. So we had to mooch around the place trying to think of things to do. So boredom is great. So I started, we'd start looking at the wildlife. I remember doing experiments with wasps, trying to, we actually sort of given a chemistry set. And so I remember asphyxiating with carbon dioxide and then rejuvenating with a pulse of oxygen, which had been created by mixing various chemicals together. And we're able to we tried testing wasps to destruction, of having them, they would go very quiet. And, then we'd rescue them before they died with a pulse of oxygen, and they would go bzzz, extremely intensely. And we'd see how long we could keep the buzz of oxygen going on for. And then we'd feed them bits of honey and things like that. So actually I sort of had a bit of a life doing that. I used to dissect termites and occasional other things. But so in fact, I was definitely evolving as a biologist when the school brought in that there was a lorry came in with one morning. It was part of their links for the university. And on the lorry were about half a dozen laborers. And there was a chap in a white coat lording it over the laborers. And on the back of the lorry was a huge tin bath. And in the bath, there was something that was clearly making noises. Well, actually, it was sort of slushing lots of water out of the bath. Anyway, the Africans very gingerly shifted this thing to the side. They the scientist, it turned out he was a scientist, a biologist, explained it was completely harmless, but the Africans didn't think so. They ran about 20 feet away and sort of shout and scream if it did anything, if it tried to raise a flipper. They said it was extremely dangerous. These animals would kill you. They could kill you by looking at you and other such things. Well, it was looking at us all and no one was dropping dead. And I thought, this is actually wonderful life to have. He'd gone and rested, he'd gone and taken it from the wild and then brought it back. And I said, what is he going to do with it now? And he said he was going to experiment on it. And I didn't know what he meant, really. I hate to think what actually happened to the animal, but it was just, I thought, right, I want to be a scientist and I want to experiment. So that was the first time I crystallized what I wanted to be, because I already experimented on wasps and playing around. So I really wanted to be a biologist. And then in my, of course, this life came to an end. I was forced to go to a boarding school as a sort of secondary school, and then didn't like it. And in the end, my parents had to come back from West Africa because my father's job was Africanized. They used to do that with the European administration, and which is fair enough. And he came and worked in England, and I went to a very good school, Newcastle upon Tyne Royal Grammar School, and they very much encouraged. We had some teachers there who very much encouraged within biology the molecular side of things. And I started to realize that yes, there were the sort of people who liked dissecting dogfish and such like, and rats, which I was a bit squeamish about. But there were those others who, but I sort of developed an greater interest in the molecular world, and in particular, DNA and proteins and such like, even at the age of 15, 16.
So early.
Because to me, this was absolutely fascinating world. And in particular, how life arose. That absolutely fascinated me at that stage.
I ordered the book you mentioned from.
Oh, yes.
I could still get the used copy. It's still on the way. I haven't got it yet. By Melvin Calvin. He also won the Nobel Prize.
Yes, he did. It was a very interesting book. It was about how it was a complete eye-opener, how molecules might come together and create life. And in those days, it was, it was a probably extreme view. I think it wasn't. I mean, now, we tend to be comfortable with that, but in those days, it wasn't a lot of. There was sort of almost religious objections to it.
Didier Queloz is also very interested in origin of
Sorry?
life. Didier Queloz, the...
Oh, yeah, Queloz, yes, of course he is, yes.
You know about it. We had a similar discussion with him, and he said that he's not only interested in exoplanets, but also in origin of life issues. So he's talking a lot. Are you in discussion with him about that origin of life?
Yes, I have. I have occasionally talked about it, not a huge amount, but I mean, essentially, he's defining the kind of atmospheres that are present on planets, which, and of course, working out, or at least taking interest with other people, mainly through other chemists, some of whom I know, who are looking into the detailed chemistry of how these different molecules may have got assembled. In other words, what you need to put together, what could arise artificially, what can bootstrap itself out of this primeval soup. So I was very interested in that. And so when I arrived at, I got a scholarship to Trinity, and I was interested in pursuing that. And I had to do a bit of physiology, which I didn't like at all. I didn't like all the live animal vivisections, what it amounted to, which I think have probably now disappeared off the syllabus. And I started to focus much more on chemistry. I had a PhD supervisor who some of you may have met, called Brian Hartley. Hartley, he was a larger-than-life figure. He was a northerner. He used to smoke a foul pipe. And my worst experience was he'd take you into his office and shut you in his office and then get out his pipe and, start smoking, and you'd end up being fumigated. I was always desperate to get out of there. Although what he was saying was always interesting. So he what I liked about, I think I look back and I realize he must have had a huge influence on me, because there's a number of aspects in which I realize I've been guided. One was I remember once saying to him, he asked me what I'd been reading that week, and he said, well, why did you read that? I said, well, it was interesting. He said, bugger interesting. Is it important? I thought, it's a good question. It's a good question. Is it important? Yes. He said, there's loads of things in science that are interesting, but does it matter? And I thought, actually, that's a good point. The other thing he did is he expected me, I remember doing some practical work. We had a project, it was mainly done in the Department of Biochemistry, and he was having a loose oversight of it. I mean, I had official supervisor there, but he was checking through the Trinity students who were doing things and asking us. We had sort of these open sessions where we'd go over things. And he said, well, I described a few experiments, and he said, well, you still haven't proved what it is you're saying. And I said, well, no, but it looks pretty much there. He said, look, lad, in science, you need to put a stake through its heart. So use some other bloody methods as well. He said, so I thought to myself, yeah, that's a good point. You need to come at something from multiple dimensions, and you need to whack them in. One thing isn't enough. You've got to put in multiple things, particularly in biology, which is extremely complicated. And so he had, I realized he had a big influence, because I decided that he was enthusiastic, optimistic, and in fact, I was really interested in becoming his PhD student, which I did. He had two projects for me. One was on enzyme evolution. Could we evolve enzymes? But this was the days before recombinant DNA. And he was doing it by genetic means. And it occurred to me this would mean getting involved a lot in genetics, which I thought was a bit much too like mathematics to me.
Which you don't
and I didn't really like the sound of it. But chemistry, protein chemistry, which was a project to look at some of the early enzymes in evolution, was sort of right up my street. That was a project which I started on, doing the protein chemistry of that. Interestingly enough, Michael Neuberger, who sadly died at that stage.
He was a great friend when I was fellow at Trinity. He was like a year or two ahead of me, and he kind of took me under his wings a bit. He helped me with many things. Of course, his work was a completely different area, but he gave me a lot of advice.
He'd actually graduated as a physicist, so he wasn't a biologist. And that's extremely interesting, because
maybe that's why he had a good connection with me. I forgot a bit the details now, because it's a while ago.
Yes, he was a physicist, and he decided that he wanted to work on that. I think he found biology more interesting. And in fact, I came to a similar sort of conclusion during my undergraduate phase. I liked the biology, but as we did more biochemistry, I began to dislike it on the grounds that I think it all seemed to me to be damn sight too complicated. Very difficult to prove anything, that life never solved anything in a straightforward way. You could never apply Occam's razor, that if you could, rather than activate something, you had an inhibitor of an inhibitor, this kind of thing. It's just kind of not, it's logical, but in fact, if you've got to think in terms of inhibitors of inhibitors of inhibitors of inhibitors, it just becomes, you're just searching through this jungle. And so I actually so my approach to biology was very much from chemistry, because I found it was a simple universe, building blocks I understood, molecules I could solve the structures of, potentially. Whereas it was interesting with Michael. So in fact, but the problem was, why didn't I go into straight chemistry? And the reason was, I felt that biology had the more interesting problems. I thought the chemists had lost themselves. I'm sure they would doubt it if there was a chemist on this call as well. But they seem to be endlessly going into molecular orbital calculations and such like. And I wasn't really persuaded that would be the future. Or at least it might be mathematics, but it wasn't, to my mind, what I wanted to do, which was experimental science. And I thought biologists, there were so many interesting questions to which you could apply the tools of chemistry. And so I became sort of firm molecular biologist, but keeping myself on the, I would say, on the non-biological side, the structural side of building new molecules and analyzing molecules, so I fully understood what they were.
The real life side of it, the real life side.
Yes.
And how to apply. And
and it's like Michael, who, he actually was very good in this jungle. I mean, it's surprising that physicists went into this jungle, and Michael had all sorts of intuitions. I thought, why did Michael think about that? Because it wouldn't have occurred to me, because I would go for Occam's razor and Michael would have a complete, you know, he used to keep lots of ideas bubbling in his head at the same time. So it's very, very interesting, I found. But nevertheless, Michael ended up being extremely successful, and we found our paths converging. He started off a year later doing a PhD with Brian Hartley. He took on the enzyme evolution project.
Okay.
I started
you dropped.
which I had dropped, which I had wanted to take. And actually he did a pretty good job on it, accepted, exposed the limitations of the methods. So I'd been right about, you know, I couldn't have done what Michael did because he was very clever and very good at genetics. He had a brain that got around all of that, very enthusiastic. But it was limited in the end because they hadn't really got the tools to analyze what was happening. And it turned out the sort of evolution that if you, as the kind of what Hartley's experiment was and what Michael did was to take a bug and effectively mutate it and just random, and then to see what, then to try to apply a selection pressure that would enable you to select for certain mutant enzymes in that bug. You'd feed it a food source, in this case, a sugar, and you'd look for the evolution of the key enzymes. Can you improve it? In fact, Michael did detect a series of improvements, but the problem was, it turned out the way the bug works, the way bloody biology works, is it doesn't create a new molecule, it doesn't create a new enzyme, it just increased the amount of the old enzyme, so the weak side activity that had got on the new sugar source meant that it could grow faster. So actually what we found was the mutations, that you just had this natural duplication process. It wasn't actually finding anything interesting about the enzyme, other than you apply a selection pressure and there's probably some weak activity which you might be able to harness if you increase the amount of that enzyme, which is, so people actually now think that evolution works quite often by a selection pressure on an organism. Then you, that selection pressure may cause you to double a gene, for example. If you remove the selection pressure, that you've got a spare copy hanging around in your genome, which then which is then free to mutate to do other things altogether.
So it gets repurposed.
Correct. So it builds in a kind of spare copies of genes, and we find that a lot of bits of genome are duplicated, and this is probably part of it. And you know, Michael was one of the first people to observe this kind of thing happening in an experimental system. At that stage, we didn't really realize that. But anyway, he then went on, he decided to go into the antibody field because he wanted to work with Cesar Milstein in the MRC laboratories. And he went off to learn about antibodies. He came back to Cambridge, knowledge about antibodies. In the meantime, I had been trying to work on developing recombinant DNA methods to change enzymes, including the enzymes I'd worked on for my PhD. And we did the very first protein engineering experiment in altering an enzyme's properties by targeted mutation at a particular site. That was work with Alan Furst, who's then became Master of Keys. But a splendid physical chemist. So I did that work, but then Fred Sanger, who'd been my boss at the MRC and a double Nobel laureate, was retiring. And Fred had been supportive of me for doing this work, but he said, look, Greg, you know, I'm retiring. Cesar will be your new boss, so you'd better make sure that whatever you do appeals to Cesar. So I went to Cesar and I had a chat. He said, well, this enzyme evolution is very, very interesting, but why don't you work on antibodies? Why don't you work on antibodies, which was his field, because we need more people to work on antibodies. And I'm sure that the things you're doing with enzymes, you could use on antibodies. So that's what got me into the antibody field. It was just luck. It was having someone who became a Nobel laureate, you know, shoving me into that field, but make it quite clear if I didn't work on antibodies, then I might not get a job. So it was you know, there was a level of soft compulsion there. I mean, he was very supportive, but he did expect me to work on antibodies, and I said, well, like what? He said, well, that's up to you to sort out. So I tried to make sure Michael and I kept clear of each other, But he put us in an office together, and in fact, Michael ended up taking one approach to making therapeutic antibodies, which was converting mouse antibodies to chimeric, taking large blocks of the mouse antibody and transplanting them. I actually decided from my knowledge of binding sites that you could actually just take some of the binding loops and transplant those. But it was a much more complicated experiment involving lots of gene synthesis, which was very new at that stage. You know, we had to buy in a machine. I had to learn how to do it. It was really quite complicated. And but in the end, we created an antibody that was much more human than the ones that Michael had created. In fact, Michael wasn't the first to do what he'd done. Other American group basically beat him to publication, which was a shame. But our antibodies were sufficiently difficult to make that even if someone had had the idea, we were able to get that out first. And that class of antibodies is now the biggest class of anti-therapeutic antibodies that was used. So that was made by taking a mouse antibody, just transplanting the bits you need into a human antibody rather than more than you need. And that way it's not seen as foreign. So that was really
you say, and you write that the body really doesn't see the mouse components anymore, you say.
Well, I don't know whether it does, but it doesn't raise significant blocking antibodies to it. Dangerous if you inject people with a foreign protein, you will make antibodies to it. We've known that for years. That's how you recognize viruses and bacteria. You make antibodies. That's what the vaccines do. They're generating a slew of antibodies to this foreign component. Whereas, for example, if you inject someone with a self protein, like a human protein, you generally won't make antibodies to it because your body recognizes that as being self. So the argument was, if we make something self enough, then we may be able to get away with it, right? So that was the idea. So in other words, it may not see. It may not see, the immune system may not see it. But we didn't know whether that would be the case. If we put it into humans, would this be seen as human or would it still be seen as foreign? So that was how we first made therapeutic antibodies from taking mouse antibodies and converting them into humans.
Maybe for time, maybe the second point is if you can talk a tiny little bit about the huge field which then led to your Nobel Prize. But I think because most of that is documented in your speech, maybe we can cut that short. And what I found is you have this very nice picture which everybody can understand with the lock and the keys.
Yes.
So, maybe if you explain that, then...
Well, okay, so the argument was we wanted to make the antibodies that you want for viruses, you can make anyway, that your body makes them. But suppose you wanted to make human antibodies to attack a human cancer cell. And they're quite often markers on cancer cells that you could recognize. So, but the issue is that you can recognize if you put it into a mouse, it will make mouse antibodies to that human protein. And therefore you can use mouse antibodies to attack human tumors. The problem is those end up being seen as foreign, and that's why we have to humanize. But could we make genuine human antibodies to genuine human proteins? Now that involves doing something the immune system doesn't do. The immune system turns off the ability to make those proteins. It's what's called tolerant, self-tolerant, so it doesn't recognize them. So we thought, well, how would we do this in principle? So we had the idea, it's a little bit how the immune system anyway works, except without in the built-in restrictions. What we did was to say, let's make a human, let's make a big library, lots of different human antibodies, and then we'll find out which ones work. And it's a bit like the question of the issue of a master thief wanting to open a lock. If you've got an unknown lock, what you could do is you could just take a large enough bunch of different keys and you'd probably find one that works if your bunch was large enough. So in fact, if you had an infinite bunch of lots of different keys, you could open any lock in the world. So the question then is, how do you create, first of all, two questions. How do you create this diversity of keys?
The library.
Right, the library. and which we worked out how to do.
You created the libraries, you created.
We created the libraries. So we created the libraries. Then we thought, okay, now we've got these libraries. We're probably only going to find 1 in a billion in there that actually opens the lock.
Yeah,
any particular lock we want. So how do we find that? And that's where we introduced.
That's a phage display. No?
That's a phage display method. So a phage display, I won't explain it in detail, but it just, it allows you effectively to rapidly identify very rare keys that open the lock. That amounts to.
Yeah. The amazing thing about the phage display, if I understand it right, is that you can have the DNA in the middle or in the body, and then also you have the display which of the protein which is encoded in the DNA. So you have both the embodiment and also the information. And you have those together.
It's what we call, it becomes a Darwinian package. So in other words, it's a sort of link between phenotype and genotype. So you've got something, a selectable marker along with the genes encoding it. And therefore, and since it can replicate, you can prepare lots of it. That was the idea. So we use that to create human antibodies, and one of which is the antibody Humira, which is the world's leading pharmaceutical antibody. Well, actually pharmaceutical product. Sells about $20 billion a year at the moment. And that was made in a little startup company that I founded called Cambridge Antibody Technology.
Okay. The other thing I think, which is also a component in your inventions, is this the concept of directed evolution. Maybe you can also say a few words about that.
Well, I mean, the issue really is it is this idea of using, I mean, directed evolution in a way is another word for Darwinian selection. I mean, Darwinian selection, you apply a strong selection pressure, and the fit will survive. And so what we can do is by essentially creating lots of variation, and by applying an appropriate selection pressure, we can make molecules do what we want. So the question is, so you have ways of creating the library, you have to find ways of having a selection pressure that selects for what you want. And that's not always so easy to devise a very potent selection pressure.
It refers to that process of using the library, and then I think you have the agents you want, you have the agents on a substrate, and then you put the phages on top and you wash away the ones which don't fit, and you multiply. And so that process is called directed evolution.
Well, we call it directed evolution, but it's just a form of Darwinian selection. It's what's happening in life. Normally what happens is organisms evolve, for example, in the soil. If there's contaminants, you will find bacteria will adapt and multiply and grow on those contaminants. I mean, over a period of time, what we were doing is we can get all of this to happen in a couple of weeks, because we can apply, first of all, massive mutational power, right, rather than relying on just random mutation, which will take years and years and years. And we can apply an extremely potent selection, because we're not working on an entire organism, we're working on one particular molecule. So we can evolve on.
How do you create the mutations? How do you create that?
Well, you can create them by putting in various nucleotide analogs. You can irradiate it. There's lots of ways to do mutations.
Oh, you really attack
it. You can copy it with error-prone polymerases. You can copy it with nucleotides that are known to be error-prone. There's no problem in creating mutants.
Oh, okay. You kind of enforce that you kind of disturb the system to create these mutations.
Correct. You disturb the system, so you can either mutate an entire gene, or you can say, actually, I know this is the most important bit of the gene. If I mutate the entire gene at high frequency, I'll probably kill it. Therefore, let's focus on the bits that we think won't kill it. So you can focus on the binding loops or the catalytic loops. You can put your mutations, random high frequency mutations, where you want.
Oh, okay. Then maybe if the next point is, one of your amazing achievements is the transfer of your research into enormously important impact in the world. And on the way to do that, one of your tools is creating companies. And when I was sitting next to you on high table in January, I miscounted your companies and you corrected me. So please, please excuse me if I
That's
get.
all right.
No,
I don't remember anything. I'm of an age where I don't remember anything now. So don't worry about it.
Do you understand that you created 3 companies? Is that right?
Yes, yes.
Yeah, that's what I have is Cambridge Antibody Technologies, then Domantis, and Bicycle Therapeutics.
Correct.
And maybe the most interesting would be if you could say a few words about Bicycle, because that's the one developing now.
Okay. Well, I mean, perhaps I'll just briefly digress with the first 2.
Oh, yes, of course, of course.
The secret with those was, and I think the same is probably true with Bicycle, was that is one of the first principles. Well, okay, so I'm basically an academic. The reason I was interested in in taking the molecules further was I could see they did have potential in going further, and I could have just handed them over to someone. But then when you've got a really good idea, and where it's like paradigm shifting, you usually find people aren't interested. So it's quite difficult to get funding. So because standard committees, like research council committees, I don't think would have supported the work at the stage.
Yeah, I want to come back to that later.
Okay. So that work was, I realized the only way to see that work taken forward was to set up a company to do it, to see that utility exploited. So I, in the position, I either could do the work, I could either just leave it, go on and do something else, having shown the principle of something. But I knew that if I did that, I would never get the credit for the work, because people would say, oh yeah, this guy Winter, yeah, he's quite clever, you know, he came up with this method, never did anything with it though. So we Americans had to come and sort it all out, and we created a company and we built this and that. So, you know, part of it was selfishness. I had no wish for anyone else to steal all the credit. I also knew there was no way I would get credit if I just left it. And furthermore, there was, I could see the possibility of doing tremendous good. And in my work with humanized antibodies, I'd met a patient who'd received an antibody, and she had a huge impact on me in making me realize, why am I, you know, I'm just this very intellectual, but we can make, you know, I'm in the position to make molecules that can make a real difference to some lovely people. And there's nothing they can do. They could talk about fighting cancer, but there's no way they can do it. You can't fight cancer, really. You've got to have some molecular help to do that. And so I guess I switched mentality from being purely driven by curiosity, which is not, I'm not saying I'm purely driven by application now, but it was much more to do with application. So I started thinking much more about, if I've got an idea, I've got to try to work out, I have a duty almost to see it taken forward. And in many cases, if it's a very new idea, you won't get anybody else to do that for you. You have to do it yourself.
Yeah.
So actually, in the case of the antibodies, that was a stage when there was no antibody market, no one was interested in it. And I had an Australian friend who saw the potential of this technology, a chap called Jeff Grigg, who sadly died, but he was quite a bit older than me. And he thought it was a really cool technology and it'd be great to see it applied. All the VCs I'd spoken to weren't interested. They said, well, Dr. Winter, tell me about your technology. And I would say, well, the idea is we create billions of antibodies and then we find out which is the one that's going to work. They said, well, how are you going to make the billions? I said, well, generally in this way. Have you made billions? I said, no, but it's quite simple. You just have to mutate and very straightforward. You can shuffle genes, but have you ever made billions? No, but it can be done. You know, just trust me, it can be done. Well, do you have any idea how you're going to select the molecule? Well, at this moment, there's all sorts of approaches we could try, but no, we haven't actually tried any of them yet. And that's what I need. Just give us the money and we'll do it. Now, that wasn't very persuasive. And they said, well, Dr. Winter, suppose you do it, then what's the market for antibodies? So I said, well, there isn't a market at the moment, but we'll create a market, a new market, you know. They said, so there's no market, well, the market actually is about 5 million. They corrected about $5 million a year or something. So we're going to millions into you for a market that's 5 billion if you capture the entire market. I said, yes, but it is going to grow. The market will grow, you know. And they said, well, Dr. Thank you so much. When you've got a bit further, when do you come back? So that was more or less the response we had, which in a way is fair enough because these are business people. And I obviously failed to communicate the enthusiasm that I had. Whereas with my Australian friend, he understood it. He was a scientist, he'd set his own company up, and he just thought it was really cool and really exciting and just wanted to invest in it. So you need to, so the key to my first two companies was finding someone with money who believed in me.
You and then being able this horse racing society.
It was, well, it was actually, it was a company that had been set up by people who were, it was a company set up to deal with the fertilization of pregnant mares in Australia. And it was a peptide company, so you can stimulate the mares and get them ready for the stallion, basically, and you're much more predictable, and it's presumably more lucrative to be able to know when the stallion, when they're ready for the stallion, and it's much more under your control. You can book the stallion and saves money. So I think this was the idea. I'm not completely sure, but I think that was basically it. That company was doing okay, but the people involved in it, the shareholders, many of them are racehorse owners or people who were in the betting industry or such like. And his major investors and board had got an attitude that was very much more of, yeah, let's give it a go. So I remember one of them, when we asked, so Jeff went to his board and explained that he wanted to make this investment. They thought his company, Peptech, should make the investment. And he said, and you know, I was told one of the board members said, yeah, okay, let's see how that boffin trots then. Let's give him some money.
So you were one of their race horses.
That's right. And then when I went out, I had to go out to Australia to meet them. And I remember them, you know, I was pretty scruffy. My hair was long, you know, hadn't trained my wife then how to cut it. It was, I remember saying, oh my God, I'm going to meet these investors. I better get my hair cut. He said, no, stay like that. You look exactly like a boffin. We want you to look like a mad scientist to be able to, because that's what they think you are. So let's just, you know, just stay as you are and just, you know, be enthusiastic and all the rest of it. And we should be able to get the money. And indeed we did. We got some money. We set up the company and it was successful. That was Cambridge Antibody. But in a way, the position was slightly similar with Bicycle Therapeutics, which is what you really want me to talk about, which was the company which
it's up to you.
No, I heard you talk about it, but Bicycle Therapeutics, I had various people working in my lab in the 90s. So they'd been through my lab, as had this chap, Jeff Grigg. I had been there for a sabbatical, so he knew me. So there was a lady postdoc called Regina Hoditz, who was from Austria. And I think she now works for Wellington Investors at that stage. So she'd done science and she decided to go into, she was quite a good postdoc, but she decided that she wanted to make some money and she went into business and got an MBA in France. And was a, you know, we would meet up from time to time. You know, she'd come back to Cambridge every year or so. We'd have dinner at High Tea. She used to come back from time to time. We kept in touch. And then she said to me, Greg, now I'm in Atlas Ventures. You've been successful with your last couple of companies. Do you know, you will tell me if you have another idea, because I'm sure we'd like to invest in it. And I said, well, actually, I may have another idea, which was the idea of creating a small antibody mimic using chemistry and using still phage display, but making libraries. But in this case, I had a great big antibody molecule. We could make a like tiny little antibody mimic, and we could use that. We could select that by biological means, and we could then use that to penetrate much deeper into tissues. So that was the general idea. And she said, well, it sounds a bit far-fetched to me, but I'll talk to the folks back at Atlas, and if I can get some other partners in with me, I will. So she actually got a series of other venture capitalists in, actually all ladies. They always to talk to each other. So we had the board at one stage was apart from me was entirely female. So there were actually Kate Bingham was one of the venture capitalists. So she's one of the people who headed the UK vaccines task force. So, in about 2010, I think they invested, Schroeder Life Science Ventures invested in Bicycle Therapeutics. So she's been one of the very long-term investors. But anyway, but in a way it was that to get going, I happened to, know people, had contacts who they had a certain faith that there was something interesting here, and I couldn't explain exactly what our market size would be, give Excel spreadsheets on profit and exit routes and things, but they could see, well, there's something there, and somehow there may be a way we can fashion this. And in fact, they gradually, took Bicycle has taken longer than I ever expected, but it did. We are now a NASDAQ listed company. We have a valuation of about half a billion dollars. And the share price has done quite well recently. It collapsed a bit at flotation, but then it's gone up as we produce news and things. So I'm learning for the first time what it's like to be a director of a floated company, because previously
I was here in Tokyo for 4 years. It's quite interesting.
Yeah. So that was how it was. I mean, it was really a matter of having people who believe in you. And it's the same as you try to gather staff. People want to work with you, and most people say, oh, it's risky, oh, I don't know, it may never get anywhere. But there's some people say, yeah, it's really exciting, let's go for it. And they're the people you want around. But it's hard. You have to have, I'm sure that level of enthusiasm that Brian Hartley had persuaded me was essential in science and actually has to take you through all kinds of negative results, was also terribly important in this case, because you just have to try to not let anything get you down. You have to press on. You just have to try to keep on going, even though the odds seem to be against you sometimes.
Maybe I have maybe 2 more points to, if it's okay with the time with you. One point is, which you made in several times at several points in your publications or articles. And one article is your long article called Antibody Engineering in Philosophical Transactions of the Royal Society in 1989, where you write that in MRC lab, antibody engineering, the potential of antibody engineering was well understood. And then a BBC journalist called John Newell found that and wrote an article or talked about it on BBC. And about the same time, NRDC rejected your applications or MRC applications. And also there was a Spinks committee in 1980, which didn't realize the importance of antibody engineering and protein engineering. So the issue here is, this is, many people will say, many governments will say, we need a national growth strategy, or we need a technology strategy. But I attended a meeting of the Swiss government here in Tokyo, where a guy called Walter Steinlin, he was the president of the Swiss Commission for Technology and Innovation. He is so responsible for innovation in Switzerland. And he said that the Swiss national technology strategy is that we don't have a strategy, but we support excellence. So on one hand, you have like the evolution, which is bottom up, which is a bit a process like you use it for creating the antibodies, no? Or this evolution, kind of directed evolution on one side. And the other side is you have a national strategy created by an eminent body and the politicians and heads of, and this is like more like the intelligent design type of thing. So what do you think about this dilemma?
Well, first of all, I just want to correct your first introduction, the history of it. It actually relates to, so it's okay. So you've got the principle. I don't want to spend a long time dealing with it, but it related to the issue with the Spinks Committee and John Newell and the BBC related to Cesar's original work on monoclonal antibodies. He made mouse monoclonal antibodies in 1975, and did not and applied to the MRC and the NRDC as to whether he should file a patent on it, and they said no. And then later, and John Newell, Cesar was a bit surprised. This is in the mid-70s. John Newell of the BBC actually read about his work and thought it was really exciting, and he was on the World Service, and it came out what a great exciting thing this would be. But that was after publication. And then the MRC, people started to get excited about it, and then the MRC and the NRDC realized they were wrong. They'd made a bit of a mistake here, and they then blamed Cesar for not explaining it to them clearly enough. So that's what actually in that thing I was getting at. I was very angry. Cesar had said to me, if any of your work, if they say it's not worth patenting, just patent it. Just try and find a way of doing it, because they'll blame you anyway if it doesn't get exploited. And it had the effect, it was very damaging to the MRC, that general representation by the Spinks Committee and by the National Research and Development Corporation, because Mrs. Thatcher was very angry that the whole field of monoclonal antibodies, which began to be exploited, was in America, and that we'd sort of lost out. And she used to harangue the head of the MRC, Sir James Gowans at that time, and tell him, you lost monoclonal antibodies to the Americans. And, and that, don't you think you can come crawling to me asking for more money for you when you're just completely incompetent, et cetera, et cetera. So
here's my handbag.
He used to be handbagged every time we'd go in there, and was very sensitive. So when antibody engineering came along, the MRC did realize the potential of it. And that was partly one of the issues I had in dealing with the Australians, because the MRC were terrified that this thing would be taken off to Australia and they would be handbagged again for letting the Australians have this improved technology. So they were twitchy as hell, but also realized the Australians might seize it anyway, and I seemed to have a good relationship with them. So in fact, they did allow me to set up Cambridge Antibody Technology, which was the very first company that the Medical Research Council set up, really, in which they had an equity stake. And they could have had a much bigger equity stake.
And I heard they rebuilt it. It was more money. They earned more money on it than the whole MRC building cost.
They certainly did. Yes. So the whole suite of patents I invented has earned them more than, it's at least £500,000, possibly $1 billion, depending on exactly how.
Overall or to the government? Overall or
no, overall to the MRC, of which most went back to the government. So in fact, the original MRC was supposed to have it all, and the government changed the rules when they snaffled it from the MRC. So it's outrageous, because they had years and years and years where they said, you can keep the money to encourage you to do it. And they suddenly said, well, hang on, you're making loads of money, we'll have that. They took it. So kind of, in fact, they made it even worse, because they said, you've got to, since you're bringing in money from these sources, you've got to predict what money you're going to get in future years, so we can take this into account when we give you taxpayers' money. And if you get it wrong, you'll be penalized. And if you get it right, you'll get the money we first thought of. And so actually, it was a scandalous thing. It involved them in lots of bureaucracy. And it meant there was no incentive anymore from the lab to do anything, the individual laboratory of molecular biology, or really the MRC, other than being beaten by government. So it was a sad story. I think it was pretty okay until the early 2000s, and then they started to snaffle the money. I mean, fortunately, I still get a tiny share. I got a tiny share of royalties, and they were going to snaffle that, but the MRC stood up and said, look, sorry, you could take ours because we're a public body, you can't take it from the inventors. Anyway, they decided in the end they wouldn't.
Yeah, Shuji Nakamura had a big lawsuit about this.
Sorry?
Shuji Nakamura, the Nobel Prize winner for the gallium nitride. He had a big lawsuit here, which made big headlines in Japan regarding his rewards for the invention.
Well, I was an employee. I expected to get some, actually at the beginning when I first made the inventions, there was no money due to inventors, but then then they did actually change the rules, so I get a very small percentage of the proceeds, which has ended up, you know, being a few million because of the sheer scale of sales, which they'd never expected. And that always actually sort of embarrassed them, really, because they'd got these rules and they'd never dreamt anyone could make as much money out of it. Because I didn't think they wanted us going off and doing our own thing particularly. They wanted us to get a bit like a bonus. But and in fact, at one stage, I talked to a treasury mandarin, and he said, well, you know, I said, look, we don't have any incentive to take things forward. And apart from making you feel it's very unfair, you're just not going to bother doing it because it ties you up in bureaucracy. You have to work on, you have to talk to patent lawyers. You've got to do all of this. And it takes, you're not given any credit for that in your research exercise. So I didn't get any credit for years for doing anything practical, because they said, we're just going to purely focused on the academic work you do. And he said, well, I suppose if it's taking you extra time, we could think of applying overtime. You could think of getting an overtime rate, but we don't have an overtime rate for scientists, you know. So we'll have to talk about that. As though I was working at a biscuit factory or car factory, you know. So actually, the whole attitude just infuriated me, and I was therefore delighted when Mrs. Thatcher sort of somehow managed to, and I think Kenneth Baker, who was then the Minister of Education, or in charge of what's now BIS, decided that he would, that the inventors deserve something. Because I went and gave my talk, and I remember he said to me, well, that's splendid work. When I showed him this patient who'd recovered from this humanized antibody, he said, that's splendid work, young man. I hope you make a lot of money from that. And I said, I don't think I'll make any money at all. And there was a stunned silence. He said, he turned to his mandarins and said, why not? He said, well, Minister, we are actually looking at this matter at the moment, and we are investigating this, and I think there'll be a report that we might be able to do something about this sort of thing. And he said, very good, I will see it on my desk fairly soon. And then he went away. And it was very funny. And one of the mandarins, this is when the mandarin turned around to me and said, why do you think you should get it? You know, what's wrong with overtime rates? You know, something of that sort. But it was just really, you didn't come into science to make money. I said, no, but I didn't come in for everybody else to make to make money out of my work. Why are all the lawyers? And it was really that annoyed me. All the lawyers and other people were charging vast fees, patent lawyers, and we got nothing, absolutely nothing to do what we were doing. And it was our idea, and we'd spent all this time.
The same problem is here in Japan. There's Shuji Nakamura, he brought that up, but it was defeated, and he's in California now, University of California in San Diego.
Well, the argument was, some people in the lab used to argue this would corrupt the lab overall. Anyway, you were asking me the bigger question.
Yeah, the bigger question is
the bigger question was, how do you have a national strategy? And I think you need both, because the strategy I took is I never focused. I never, if someone had told me you need to make some therapeutic antibodies, just get on, find a way, I wouldn't have done it. As it was, I followed my own ideas, and therefore I felt I got ownership of it. And I wasn't there to be told what to do, because most scientists are very bullshit. You're not going to be pushed around, someone tell you what you've got to do. You want to do what you want to do. So I actually felt that in my own research, I wanted to, I was actually interested in therapeutics, although it was not within the normal theme of things being done in the laboratory. I was an outlier in the laboratory. Most people were very, very academic. So it was, but I know if they'd said, you've got to do this, you've got to do that, I wouldn't have gone along with it, and I would have probably left. So I don't think you can, it's always a good idea to tell people what to do, because intrinsically they don't want to be told. The people who are good at science and go into science don't intrinsically want to be told what to do. You've got to have some faith in them to do things.
I mean,
on the other hand, and so the work that I did, I actually applied things because I felt I had a moral duty to do so. So I did some work. I realized there was some potential of the particular kind of protein engineering I was doing. I could see it could be used to make humanized antibodies. I could see the bicycles could be used to make mini antibodies. It actually had arisen from another bit of work that I don't explain, the Nobel thing. And it was more by following other directions and realizing here's something that we can capitalize on. So discovery, an idea that may have applications beyond what I first thought about, first reasons I did it. And I always felt scientists should have a duty at least to draw someone's attention to that. And I did. And usually what happened is no one was interested in it. So I thought, well, actually, I think it's really quite a good idea now I think about it. I'd have to do something myself, and I've done it.
3 times at least.
So it's more a question of opportunism, of taking advantage of having mechanisms that allow people to have the freedom to do what they want, but where there are things that are potentially applicable to help them, or in fact to facilitate it or do it if they don't want to get involved themselves, to do it. So that's what you need. That's the first thing. But on the other hand, when I set up Cambridge Antibody, after I set it up at a very early stage, the MRC came to the view that in fact, this antibody engineering I was doing and making these big libraries was an important strategic area, and didn't manage to get money from the government to set up something called the Center for Protein Engineering, which was to actually support my work and some work Alan Furst was doing. He was doing work on enzymes, I was working on antibodies. And we got a significant sum of money, which meant that my group in the lab, which I'd only had a group of about 6 people, at one stage I expanded to 24 people to develop this technology. And in fact, at the same time, the company was doing stuff, but we only had about 4 people in the company being paid by the Australians. So actually a lot of the grunt work was done, and by postdocs quite often who came on their own funding, and to my lab, I had lots of equipment, I had all the facilities for them, and that was funded by the MRC. I had space to put them in. And that work from the Center for Protein Engineering, where we made lots of large libraries and we showed the principle of how you'd select them. And then what we did in the company was the company dealt with all commercial approaches. So it dealt with approaches from pharmaceutical companies that wanted someone to do stuff for them. We said, go and talk to the company. We just developed the technology, which was what we did in the lab. Now that investment was really very important. That I don't think we could have gone as far without that investment from the Center for Protein Engineering. Now, the Center for Protein Engineering was part of a strategic initiative set up again by Mrs. Thatcher called interdisciplinary research centers, where you bring a number of people together and you around an expert in the field or some experts in the field, and you try to you give them money for a defined period and you just let them get on with it. And you expect, in fact, you expect them to have links with industry. So we were actually, so it was rather different. In the laboratory of molecular biology, there was a little bit of a keep off for industry. They weren't really, in those days, fantastically welcome. The Center of Protein Engineering, it was in our mandate that we had to get involved with industry. And therefore, in fact, immediately I could tick boxes by saying, yes, I'm a director and I'm a founder of Cambridge Antibody Technology. That meant I could cover that part of my brief by Cambridge Antibody Technology, the company I'd set up. Now that model showed to me, now the IRCs were set up by Mrs. Thatcher as a strategic initiative. So the idea was you'd identify strategic areas that could be important for the United Kingdom, including, in this case, the identified protein engineering could be important. And that was, I think, important to act as a nucleus of technology for Cambridge Antibody Technology, which in fact harnessed the results we got. So I think it required both. So I think that in some cases, you can have a strategic initiative around an area, and that's very useful to have public funding to say, look, these areas we think are going to be good for the national wealth, let's put some money into them. There are other areas where you just need to rely on opportunism and you need to facilitate it, but to make sure that at least the taxpayer gets to return, as happened in the case of my companies. And there's probably other stages where you may even want to have a more closely directed program of research. I don't have it in my own experience, but I dare say that, like in the war, there were things we had to do with development of radar, etc., where you needed a closely directed program of research, absolutely direct to an aim, which was publicly funded. So I think there are many different routes, and the main issue is don't be over prescriptive allow these roots to develop, allow them to evolve, because they can all be good.
Keep flexibility.
Yeah, keep flexibility. And actually, that's the important really thing about evolution. So we come back to evolution. Evolution works by creating lots of variation and then selecting those that work, right? So actually, we need to do the same thing. We need to, and sometimes an organism will be very successful, and other times it's past its sell-by date, and you need to have other organisms. So actually, in my view, you want a vibrant ecosystem with lots of different approaches to funding science.
Well, you have no path, no?
Yeah, I've had grants go in, for example, some to Cancer Research UK, not from me, but from people coming to work with me from Cancer Research UK.
Yeah, yeah.
And at the same time, they've also applied to the Medical Research Council. And I can have on some point, a damning report from the Medical Research Council on it, back on this poor postdoc's application, and a brilliant thing back from Cancer Research UK, which does see the point of it, and vice versa. I'm not saying any, but so the process of evaluating research and looking for potential, extremely difficult. And I don't begrudge those committees. We all make mistakes. I'm quite sure I've made many bad calls on what's good, what's bad. I've been slow on some things, much faster on others. So we all make mistakes, and on committees, there's a tendency to focus on, to come to a consensus view, which actually is usually the most boring view, rather than the more individualistic approach. So I'm actually keener, I would say, in a series of different vibrant ecosystems, all competing with each other. I'm basically a Darwinist. I believe in competition and funding. I'd like to have multiple bodies. I'd like to have the ability to apply to those independently, and we have that to some extent in the UK with some Wellcome grants and some MRC grants. And
the UK system is very, very good.
Silos between them. I mean, I discovered many years ago, there were even invisible silos you didn't know. So I remember Women's Club Agreement. Now, to my mind, that's completely unacceptable, because everything should be transparent, and I think things should be competitive. So I'm very much in favor of competition, and in that sense, selection process. There's different selection committees, and some get it wrong, some get it right. It's the same as applications to Trinity. Some we get wrong, some we get right. I mean, it's just, and you just gotta bear that in mind. You make mistakes. Maybe
because of time, if you, how is your time situation?
I'm fine, carry on. I'm sure your time is much worse than mine.
No.
It's my morning. I'd be falling asleep.
Friday evening. If you don't mind, I have one more question on my list, which is about COVID, because that's often related to a lot of your work. And then maybe we can take one or two questions from the audience, from the people present here, if you like to. Is that okay?
Sure.
So
yeah, it's fine.
You have, I found there was on YouTube, there's a GAP Summit December 2020 interview with you where you explained about COVID. So maybe it's not necessary for you to repeat what you said there, but if you have a kind of short, if you can say something to the relevance of your work to COVID directly.
Okay. Well, actually, I have not the faintest recollection of what I said. And that's,
okay.
I don't know. You're probably better. You're more knowledgeable than I am. So I don't know what I said. So
you answered the different areas of.
So I could just say, look, the areas of the kind of things I'm doing are potentially relevant in probably two ways. Mm-hmm. The first, less relevant, actually. The first is antibodies. So antibodies can be made to against the COVID virus and can knock it out. Right? But the problem is the virus changes, then the antibodies don't bind. And so at the very beginning of the pandemic, we were surprised to see that the virus, although it's an RNA virus and would be expected to mutate, was not mutating as fast as we'd expect. And that got attributed to the fact that the virus has what's called a proofreading polymerase. So the virus, most RNA viruses, flu makes, and many other RNA viruses make mutations because they don't, the polymerase, if it inserts the wrong nucleotide, it doesn't have a way of correcting it.
Okay.
Our own way our own DNA is copies, there's proofreading steps, which mean if it inserts the wrong thing, the body has a way of recognizing the wrong things there and cutting it out.
Mm-hmm.
But viruses quite often don't, RNA viruses quite often don't have that. And flu doesn't have it, but it turned out that SARS does. And therefore, when we saw this thing was relatively stable, seemed to be relatively stable in the early days, it was assumed that this was because that it was hopeful for making antibodies against. I was a bit doubtful, but nevertheless, it seemed to me possible that if the vaccines didn't work, and perhaps for some people who didn't, vaccines were always going to be cheaper, but if perhaps for some people who couldn't vaccinate or wouldn't make a good immune response, it would be a good idea to give them a shot of antibody. And I remember looking into that in quite some detail. But as the pandemic has gone on, we've suddenly found some major mutations occurring. Of course, the reason for that is that the virus is adapting to the population. At the very beginning, where it's ripping through a population that's not seen it, it doesn't have any antibodies. There's no selection pressure. It could just rip straight through. And therefore, it manages to be propagated perfectly well in the form in which it's in. And therefore, there's no selection pressure. So although there's, whatever the mutation rate is, there's no selection pressure. It's not going to change hugely. But the minute you start getting a population that's substantially infected, or for that matter, gets given a vaccine, you are going to, the virus will mutate in order to be able to escape that. But that's one reason why, having started a vaccination program, in my view, you want to finish it as fast as possible. Because the virus will evolve to get it through. So this idea, well, we should ship, we should do 10% of the UK population, then give all the rest away, is a mistake in a number of ways, because it means that you will end up with a large population of people with the virus mutating furiously within those population to the response that people are making. In the end, it will come back and get the people that you've got. So this is the problem with the virus. So anyway, so my own view is antibodies are probably not great, but certainly it's given me some insights into which I'm quite sure aren't unique. I'm sure other people have them, into what's going on at the moment with the virus. The other thing that we are looking at is it may be possible to make these bicyclic peptides to act as some kind of therapy. And there's two ways you might want to do that. You might want to try and stop the virus being able to latch onto cells, and you might want to perhaps alter or block some of the host proteins which allow it to come in. So there's two sort of ways you could try to do this. And so everyone's looking at this, and so Bicycle Therapeutics has also taken a look at it. And the argument is that it may be that whereas the virus, it's well known, for example, a virus, the antibody binding site, for example, blocking a virus from binding, is quite large, and therefore one small mutation in the virus can throw off an antibody. So in other words, it's just imagine you've got two large entities. A protuberance in one can stop two surfaces being able to come together, and therefore you don't get any binding. It's a bit different if you've got a much smaller target. If you can home in on a much smaller part of the surface, you may find that actually it's much more difficult. The virus has got to make a mutation just at that point, not at a whole number of possible points. And therefore, by reducing the target area, by making, for example, a bicycle, which is a much smaller molecule, which is only binding to part of it, perhaps a much more conserved part that the body can't change so much, that it may be possible to create something that would block the virus attack. So Bicycle, I'm quite sure along with other companies looking into this is at some level. We have some funding from the government, a small amount of funding to look into this. Whether it will be possible, whether anything will come out in the near future, I don't know. There's probably other ways as well. For example, we are looking at ways of stimulating the immune response.
This is to overcome this overreaction.
Yeah, well, you can look at ways of dampening down the immune response, and people have made antibodies to dampen down the immune response, and we've also looked at bicycles to dampen down the immune response. You can also look at things that would activate the immune response, but at an early stage. So actually the real problem is you need antibodies and you need an immune response early on, but that response can become overpowering later on. So actually you may need to stimulate in the beginning and suppress it later on. So it depends exactly where you are in the course of the disease. Mm-hmm. So those are things that we can, with these selection technologies, we may be able to evolve molecules that do it. The problem with, I don't know whether we will really end up doing very much because it's extremely competitive. There's so many people working on different ways of looking at the virus that really aren't enough patients to test it out on. Setting up clinical trials takes absolutely ages. Mm-hmm. And I think that in terms of cheapness, essentially the vaccines, particularly the RNA vaccines, are probably about the cheapest way you could do it to get mass coverage.
Yeah, you explained that your therapeutics are at the high end of the prices normally.
Absolutely, the high end. So you only want them with patients who are going to die otherwise.
Really need it. Yeah.
Yeah.
Thank you so very, very much. If you still have a few moments, maybe we can. Sure. Maybe we can ask if there are one or two questions from the people attending here. If you could send them via the chat here, that would be great. If anybody has questions here, maybe you can either speak out or you can send them via the chat.
Hi, my name is Evan. I just put it in.
Hello.
I just asked. Thank you, first of all, very much for the talk. I'm in Boston, so I had an early morning. My question was with regards to your experience about starting the companies. First of all, congrats for all the work that you've done. What was the experience for the private companies versus the one that's like floated? Like, why go to a public market where you're subject to public scrutiny and probably, like I work in investing and I don't know two things about some of the companies to the level that the scientists in those companies do. And I think the expectation is also that I won't, or at least I hope so, because otherwise they're going to be vastly disappointed outside at least the fields of my study. So curious about your experience with that. And
well, experience, I mean, it's really hard to know what's best. I mean, my own view would be it's best to keep a company private for as long as possible, because if you can, but of course, if you've got venture capital investors, they do want an exit, and the flotation and going public provides an exit. It also allows you to bring in much larger sums of money than you would otherwise bring in by going through the sort of private. There's a larger pool of public investors who can invest in public entities, but that's not the case with privately. So I think it gives you more money if you've got a business model in which you're generating income. So you're able to generate an income stream, then you may be able to support your own research at your own expense by selling something or the services or whatever. For some of the things that in fact, for all the things I've been involved in, it's not possible to immediately turn that into an income stream and be able to stay ahead of the competition. So we needed to have money in, usually nothing like a standard American biotech company would get. So it's actually much more difficult to get money in the UK than it is in the US. In fact, one of the reasons that we floated in the bicycle therapeutics in the US is because the markets there are bigger, the investors got much deeper pockets. They're more attuned, and so we went on NASDAQ. So in the case of Domantis, the second company I set up, that was purchased. The exit was by purchase. I would have actually liked to continue it without being purchased. We got an offer from GSK, but the thing is with venture capitalists, if you wave a large check in front of them, they just can't stop themselves drooling and just taking it. So they had a difficult choice, because we were at that stage. We were going to need significant monies to take our program of therapeutics forward. And so they either had to shove in tens of millions or sell it for 230 million. So in the end, they thought, oh yeah. So basically, they hadn't got deep enough pockets. So one of my, when I set up Bicycle Therapeutics, I thought I'm going to go to venture capitalists who've actually got decent pockets, and perhaps get several of them involved. But that itself ended up being an issue, because if you've got multiple VC funds on your board, it's, I would say, I mean, the experience has been a bit mixed. We did list. Some of them are still, actually, most of them are still there, still investing. I think all of them have got some investment still left. Most of their investment still left. But they did want to have an exit. That's all sorts of, their fund has a termination date and all of those things, which I don't really understand. So they do have to get out. They have to persuade their partners that, they can't just keep on pumping money for 10 years. It might take you 10 years to be able to get to a position where you can get an exit. So I've actually now forgotten what the question was. I was sort of went back down memory lane thinking about the different problems we had. But I'll stop there. And sorry, if I haven't answered the question, whatever it was, just ask again.
No, that's great. Thank you very much.
Thank you. We have two more questions, and then maybe we'll leave you to your next Nobel Prize. The next question, the second question, is Andreas Demetriades from Edinburgh, I think, with a beautiful castle in the back. Andreas.
Thank you, Gerhard. Thank you, Sir Greg, for sharing your story. I mean, it's great to hear that you started as a young pupil who liked biology, and then a young scientist on novel ideas, and then through the evolution of venture capitalism and university spinouts. But one of the things that stood out when you were describing yourself is the word, I'm an outlier. And I like that, but I feel it comes with a sense of pressure. Of course, if we can refer to Malcolm Gladwell's book on outliers and the sensationalization of things. However, how did you reconcile the internal pressure of being an outlier with presumably more pressure to also conform to other ideas, institutional or professional or other things like that? I hope that makes sense.
Yes. Well, no, I understand. I think probably I have a thick skin. So therefore, if I think I'm right, I'm not going to worry too much about what other people say. I haven't been on any, I refuse to go on any unconscious bias courses and such like, or I've had pressure at all sorts of institutions to do so. So I can resist pressure. I would say the biggest pressure to me was not the opinion of people around me or the feeling that I was an outlier. It was more that I was worried about being able to deliver my mainstream research. In the MRC Laboratory of Molecular Biology, there's a sort of general large fund, which is a block grant, and it's distributed through the heads of the division to the individual scientists. So we don't have like a specific grant for a specific piece of work. You sort of had to, I'm now retired from there, but we had to justify what we were doing every 5 years and put in some light plan of what you do in the future. Now, as time has gone on and more bureaucratic, it's become more difficult, but early on it was very preliminary plan of what you wanted to do, and they really judged you on what you'd done as being basis for going forward. So to me, I was at constant anxiety, which to me was more pressing, of being able to deliver what I was something at the end. I was always worried about opening up a journal and finding that someone had got there first. Just constant horror of this kind of thing, and also the horror of referees mucking you about. We produce a perfectly decent paper and they want yet more and more data for no good reason. It's not based, just because to obstruct you, just
sometimes it's just competition, no?
It's competition. It's competition. And some people, they're not even competitive with you, but they just want to in my view, they just want to be difficult.
Slow you down because
some people are just very pedantic. They want you to do absolutely everything. You say, well, it's not relevant to the main theme. I mean, yes, of course I could do this, I could do that, but that's a straw man you've set up. Look at the abstract. What do the abstracts say? This is what I'm claiming. And the rest of it, yes, there's all these side issues that we've opened up, but actually I don't want to go, why do I have to explain what every bloody side issue is? I just want to, perfectly happy to explain what the main issue is. So that was, to me, the real anxiety was all the time being scooped and also not being as good as my peers in the MRC, who are all working furiously in their own area. So it was a constant feeling that you could be seen to be the weak link, or it's internal pressure. And that, to my mind, was much more stressful than any feeling that, well, I didn't fit in. In fact, many people feel they don't fit in. I didn't really fit, even though I became part of the establishment in Trinity, in the MRC laboratories. Even though I was deputy director of the place, I still didn't feel I fitted in properly. And it didn't really matter. People said, well, that's Greg, but they had a tradition of appointing all kinds of eccentrics and misfits as being the head of the lab. And so it didn't worry me too much, because there was a tolerance of people who were difficult, opinionated, sometimes wrong, because actually, broadly, they were a good thing. And they were standing there for the institution. I tried to stand up for Trinity. I'm quite sure I didn't agree with many of the things that were being done. But, I did, you do your best.
If we can, is that okay, Andreas? If we can go to the next question, is by Professor Fukumoto from Kyushu. He's the mathematics professor from Kyushu.
Thank you very much. I enjoyed your talk. And Professor Tasuku Honjo won a Nobel Prize as the same year.
Yes.
And as I understand, the cancer cells has a mechanism to make invalid the antibody function. Then Tasuku Honjo found how to stop this action. And I want to know, what is the relation or difference between your method of getting antibody and Honjo Tasuku's work?
Right. So my method is I'm technologically driven. So I'm involved in making the kinds of antibodies that get used by Honjo and others for all kinds of applications, including rheumatoid arthritis, attacking cancer, etc. What Honjo discovered was that in tumors, there are tumor cells will, in principle, should be recognized by part of your immune system, which is called T cells. So T cells is a cell which will kill tumor cells. And one of the surprising things has been is that people discovered that and we always assumed that the reason that your immune system didn't chew up the tumor was because the T cells weren't getting into the tumor. But then it was discovered they were getting into the tumor. In fact, they were there in quite large amounts. It's just the tumor had turned them off.
Oh, yeah.
And what Honjo discovered was the mechanism, one of the mechanisms by which that occurred. And he found if you make an antibody that interrupts that mechanism, that you can now free the T cell to attack the tumor cell. Mm-hmm. And therefore, this is what these so-called checkpoint inhibitors. So Honjo and James Allison, the Americans, so Honjo discovered one side and Allison discovered the other side of this pathway. It's a very important pathway because it shows it showed actually a whole class of receptors, which mean you can turn up T cells up or down. And in fact, deactivate the T cells. And for example, in Bicycle, we're looking at ways in which we can directly activate T cells, but because these little bicycles are short-lived, we could titillate them, and then I can either continue them if it's safe, or we can just turn them off and they'll just go through the kidneys and be got rid of. So we're looking at those kind of mechanisms. And we're also looking at titillating them just in the presence of tumor cells. So they won't get activated unless they're actually inside a tumor. Our work actually now does sort of come into the areas that Honjo, who is a fantastic scientist, I have to say, has been working with antibodies for many, many years. He was involved in a lot of the early work with the antibody genome structure and such like. And in fact, the work that we did, we were able to, Honjo and co sorted out all the genes and how antibodies are put together. We essentially took those pieces and used those to create new molecules, of therapeutic molecules, which actually later Honjo ended up using in his work, that kind of engineered antibodies.
Antibodies were, I think, partly discovered by a Japanese scientist in Berlin, 1880 or something. I forgot the name.
1890, Kitasato.
Kitasato, yeah.
Kitasato, yeah. Behring and Kitasato.
I think he was
German, so Prussian, and Kitasato he was discovered in Behring's lab. I think it was in Behring's lab. I think he came to work with Behring and then went back to Japan.
in
Behring.
Berlin. Maybe we take the last. Is it okay? Last question I have
Yeah.
from Miss Su Chan. Su Chan, are you still
Right. Thank
hello.
you, Greg, for
could you
organizing the
introduce
talk and for taking the question.
Could you introduce
Can
yourself?
you hear me?
Yes, I can hear you. Yes.
Okay.
Could you just introduce yourself, what you studied at Trinity and so on, because I've never heard about you and maybe the others.
Right. Very quick, because it's not about me. I have a real passion for chemistry, and I enrolled to read chemistry, but ended up doing economics back in 1991, chiefly because I couldn't see myself spending my life in a lab coat. But incredibly sort of respectful of the work that's gone on, and certainly now been with BlackRock, Schroders, and Allianz, so very familiar with Humira and Abbott, and also following Bicycle Therapeutics on the Nasdaq. My question was staying on the topic of COVID and the vaccine rollout. It's gone some way in the UK. Not so far, in other areas, clearly beset by constraints in terms of making or scaling up the production of these vaccines. I just wanted to know how you see the evolution of this virus. I mean, would the vaccine be predominantly the way out, or would there be other sort of therapeutic solutions required? Because there is just that chance that we're not going to roll out the vaccine fast enough to circumvent or to we're going to be behind the curve for in terms of mutations. So
I think we will be behind the curve for a while. We will be behind the curve for a while. I think that there's in the end, we will evolve together with the virus. The population will evolve. Its immune systems will evolve. So the virus will gradually reduce in its severity. What I think we should expect to see, people have been infected with SARS-type coronaviruses for years, and we've adapted to them. So I think there are a number of things that can happen. So if the virus mutates, the spike protein, which is the key thing people are using at the moment in vaccines, will your antibodies will no longer recognize the spike protein if the spike mutates extensively enough. And the virus will see if the virus is different, it doesn't have an existing antibody response. But on the other hand, it will have seen if you've got other viral proteins in there, and in fact, some of the internal parts of the spike protein will be recognized by T cells. And so the virus can't change everything. It's not going to change its central core, and that central core will be recognized by T-cell immunity. Now, T-cell immunity is these cells which come along and direct. What T-cells would do is they would kill infected cells, and they don't attack the virus directly. Antibodies attack the virus. The T-cells will kill the infected cells. So you've got one layer of defense that's being established, from one infection to another infection, even if the virus changes. In fact, one of the arguments, I'm not sure I think it's true, but that children, for example, I think there's lots of reasons why children don't seem to suffer as much. Could very well be they've been more recently infected by influenza. By cold virus, coronavirus, of a different species. And in fact, there's some common proteins in there in the virus, which actually therefore means they've got some pre-existing level. Now that's one point. There are other reasons as well, like they have a more active innate immune system, which actually responds more ferociously in general than initially at the beginning than the case would be the case with old people. So I think that what I would expect to see is very gradually, the virus will continue mutating. We will get new strains coming in. For example, the South African strain, we will probably find that we get some protection from it. The real issue is to turn this virus into something that is not going to kill you, or let's say just really kills you if you were going to die anyway. In other words, if no worse than getting a bad cold. So I think that it's important to establish it, to move as fast as you can with vaccination, but also, of course, as people get infected, they are establishing immunity to that strain and to other strains to come. So what I would like to see personally would be more attention made in the vaccination program to incorporating other COV2 or coronavirus proteins into the vaccines. In order to stimulate T-cell immunity, I think that would probably be quite a good idea. Now, of course, there are some that do that, like the Chinese ones, which are based on an inactivated virus. There could be some advantages, but there's no reason why you couldn't do the same thing with the Oxford vaccine. You could start putting some other genes in there. The same thing with the RNA vaccines. You could start putting some other genes in there. My own suspicion is it would be quite a good idea to start sticking some of the genes that would help you with the T-cell response into vaccines. And I'm sure this is being done. So at least I hope it should be being done. So I think we are going to face a problem with the virus will continue to go through populations, but it will kill fewer of us.
And there might be new waves of new viruses coming in the future.
Well, there's obviously a completely new virus. To my mind, what we've learned from this is how, several things. We need to be able to move much faster in creating new vaccines. In my opinion, the fastest way of doing that would be to use RNA vaccines because they are much faster to put together. Mm-hmm. And all you need is to have loads and loads of polymerase to be able to make them, some fat and some few other bits and pieces. They're a purely synthetic vaccine. And you need to be able to, those who've got much more chance of rustling up. If you've got to grow cell lines and propagate them, it really does take a while. On the other hand, it's cheaper. Mm. But I think the RNA vaccines can be brought down in price. With any new technology, the price will, in the end, come down. I remember many years ago, I did a stint in industry, and it was in Procter & Gamble soap granules section, and they said they were looking for a sequestrant, and they wanted me to look into possible sequestrants that you could use in detergents, just like a literature survey. I remember looking at it, and I also could. They said, look at the price as well, but don't worry about the price. They said the major ingredient in DAS is actually sodium tripolyphosphate. They said when that was first introduced, that was like gold dust. Actually in the end, industry could scale up to it. So I think we don't want to worry too much about the price at the moment. I think RNA vaccines Particularly, there are vaccines which can so-called self-amplifying vaccines, where the vaccine can actually, the RNA can go into the cell and can be amplified in there, giving you an even bigger response, and you perhaps need less of it. Almost certainly, they will find ways of delivering that without, at room temperature. I don't think that's an insurmountable problem. It's just a question of stabilizing the little fatty globule and then proving that it's stabilized. But of course, when Pfizer and Moderna made the vaccines, they didn't have time to test all of the conditions, and so they just had to go with the most conservative conditions. They could choose where they knew it would almost certainly work. They didn't try extending it or trying to extend half-life. I'm sure they're all working on that, and that will come good in the end.
So much, I think we have already taken up so much of your Friday, probably noon now in England.
No, it's Friday noon. No, it's quite fine. I'm very happy. I'm very happy to do it.